All right, hi there. This is the walkthrough video for Thursday. It is November 7th in Try 2 Week 1. This is the question that we hoped you could answer. A protein's blank, which is specified by its gene sequence, allows it to do its function. What do you think it is? The answer is shape. A protein's shape, which is specified by its genes sequence, does indeed allow it to do its job. So today, if you were in class, we did a lab about enzyme shape and how it affects reaction rate. It was a really exciting time, and we tried to change pH, which changed how the protein's amino acids do interact with each other. So where have we been? We know from yesterday's assignment that DNA is absolutely essential for living things. And we learned that it specifies the real life properties of enzymes. And now remember, we're using an actual enzyme called catalase. So today we're going to pit catalase against hydrogen peroxide. Hydrogen peroxide is used for cleaning wounds and cuts and scrapes and first aids. It's toxic to living things, which is why it can clean wounds out of harmful microbes. But hydrogen peroxide is made by living things through chemical reactions that are necessary for life. So it's toxic, but like your cells also make it. Enter catalase. Catalase is an enzyme. It's made of protein. Catalases are found in living things from bacteria to potatoes to people to yeast, or at least enzymes that do the same job, even though they might not have the exact same shape or structure. And catalase denatures hydrogen peroxide to not harmful water and molecular oxygen, right? Now we know that shape is important to protein function. And the question here is, how can we change catalase's shape? Well, one way to change shape is mutation, a change in the DNA. So here's a typical gene, you get a typical protein, it does the typical job. If you end up with a DNA change, you can end up with a shape change in the protein, and that can mean some really big medical interventions are necessary for people, and they experience negative consequences that we might think of as disease. There's another way to do it, though. You can change shape in the lab of a protein by changing pH, how acidic or basic something is. So here's an enzyme. It's supposed to bind this substrate and break it apart. If the pH is correct for this enzyme, the reaction is going to proceed and you get the products. But if there's extreme pH, if there's a lot of pH changes, the site denatures and you can't get this stuff to bind. Here's another example. Here's the amino acid chain that makes up the enzyme. All the little pieces are in the right spot, and here's the active site. This shape can bind exactly there. If you change pH, it breaks the forces that hold the enzyme together and the substrate no longer fits, right? So if the enzyme is like the one up at the top, it's going to be able to do its job. If it's like the one at the bottom, it won't be able to bind the toothpicks, the substrate, right? It won't be able to do that, and it will not be able to catalyze the reaction. So we utilize the fact that this enzyme makes oxygen gas to float discs that have been soaked in the yeast solution. So these are the instructions on the assignment. We talked a little bit about the data table and graph, and then we did some cleanup instructions. Hopefully you are able to remember the word enzyme. That's what goes here. All right. Hope that went fast, but I hope you have some idea of what we did. You can look through the paper. You'll need the paper even if you were absent today. All right. Take care out there. We'll see you soon.